In today's lecture, we will talk about Golgi apparatus. So first of all, we will discuss the discovery of Golgi apparatus. Golgi apparatus was firstly observed by George in 1867 and firstly discovered by Camillo Golgi in 1897, which was Italian scientist. Now, let us talk about who named this originally. The name given by Camillo Golgi in 1898, and Golgi apparatus was named in 1910, and Golgi complex was named in 1957. So, how we will define Golgi apparatus? A network of membranous structure present in cytoplasm. So let's talk about the occurrence of Golgi apparatus. These are present in all eukaryotic cells except mature RBCs and mammals, pseudotubes of pteridophytes, and absent and male or female gametes. The size and shape of Golgi apparatus. Size range from 0.5 micrometer to 2 micrometer. The shape of Golgi apparatus is pleomorphic shape, change shape according to their functions. So let's talk about the structure of Golgi apparatus. The ultrastructure of Golgi apparatus was proposed by Dalton. So it is also called Dalton complex. There are three parts of Golgi apparatus. First one is cisternae, which is tubular in shape and 1 micrometer in diameter. The second one is Golgi vesicles, which is spherical in shape and 300 nanometer in diameter. The third one is Golgi vacuoles, which is spherical in shape and 600 nanometer. And there are two phases of Golgi apparatus. First one is convex phase, which is towards the nuclear membrane, and the second one is concave phase, which is towards the plasma membrane. Now, what are the chemical compositions of Golgi apparatus? First one is lipids 40% and second one is proteins 60%. Now, what are the main functions of the Golgi apparatus? Number first, packaging of materials received from endoplasmic reticulum. The second function is secretion of materials. 3. Take part in cell plate formation during mitosis. Number 4. Secretion of medulla.